Hello everyone, in this video we are going to see what exactly are exons and what are introns. Exons contain the actual genetic information coding for the protein. That is, they contain the genetic codons required for the polypeptide chain synthesis which determines the amino acid composition of the protein. Introns are intervening non-coding segments of DNA. They have to be removed and exons has to be joined to make mature mRNA for protein translation. Different exons are frequently combined by alternative splicing to produce a large number of unique protein. From the same primary transcript mRNA or heterogeneous mRNA via the alternative splicing, that is the splicing and random joining of the exons in a disordered manner makes the unique proteins or different proteins, variety of protein products from the single heterogeneous RNA encoded by the same gene. Examples are the transmembrane versus secreted immunoglobulin. They are encoded by the same gene, but due to the alternative splicing, after the removal of the introns, the random associations of the exons made these two different types of immunoglobulins. Same with the tropomyosin variants formed within the muscle cell. Tropomyosin is encoded by the same gene, but the alternative splicing brought the difference and made different types of tropomyosin variants. Dopamine receptors. There are 5 different types of dopamine receptors found in the brain. So these differences are due to the alternative splicing, even though they all are encoded by the same gene. So alternative splicing brings the small differences and make unique protein forms encoded by the same gene and helps in different cell signaling and different cellular processes in the cell as well as in the human or eukaryotic organism. Introns are intervening sequences and stay in the nucleus and removed by the spliceosome during splicing, whereas exons exit and these contain genetic codons required for polypeptide chain synthesis. Variants in which splicing occurs abnormally can lead to the diseases such as beta thalassemia, Gaucher's disease, Tay-Sachs disease, Marfan's disease. So if the splicing sites are modified because of mutations or the mutations taking place at the junctions of introns and exons may change the splicing region and spliceosome may not be identifying the region, producing or leading to the formation of an abnormal mRNA which encodes abnormal proteins and leading to the different disorders such as cancers, beta thalassemias, Gaucher's disease, Tay-Sachs disease, Marfan syndrome. So let's look into this picture and find out what is alternative splicing. So as you see here, there are exons and introns. The introns are removed during splicing and random joining of these exons. Randomly the exons are joined and made different proteins from the same heterogeneous mRNA. In the first mRNA, there is third exon is missing, and in the second mRNA, mature mRNA, the second and fourth exons are missing. So this is brought by the random associations of the exons, random associations or joining of the exon regions, making the different variant forms of the proteins, different variants of the same proteins having different functions and roles, or they may coordinate with each other and help the cellular signaling or cellular processes.